Hello everybody, I'm Stefan Christen. I'm a metabolomics scientist at Nestlé Research, and today I want to speak with you about our latest research, which is about NAD boosters and their differential modes of actions in humans. Just a quick disclosure, I'm an employee at Nestlé, and all the results that I'm going to show you was conducted in very close collaboration between Nestlé Research and Development and Nestlé Health Science. So everything starts a bit with mitochondria. Mitochondria are the power plants of our body, so you have it in most of your cells. And they are very important for energy production itself, so they host the TCA cycle, or they are there for fatty acid oxidation. But they are also important for redox homeostasis, calcium homeostasis, regulation of apoptosis or cell death, or also regulation of inflammation. However, as we get older, we have a decline in mitochondrial content, but also in mitochondrial function. And this is generally considered as one of the hallmarks of aging. So what does mitochondrial dysfunction mean? So mitochondrial dysfunction is tightly connected with inflammation, with aging, and oxidative stress. So you have a disbalance in energy production, you have oxidative species, so-called ROS, that increase. You might damage your mitochondria, you have disbalance in cell death. And finally, these dysfunctional mitochondria, they are linked to several disease states, so it can be impairment of muscle function, for example, sarcopenia. You can have chronic inflammation with age. You can have kidney dysfunction or chronic kidney disease, cognitive decline, so there were many links with Alzheimer's disease or Parkinson's, and cardiovascular disease. So given that we have all of these effects and it's all around energy metabolism and mitochondrial biogenesis, we thought there might be a very good opportunity for vitamin B3 or its derivative NAD. So NAD, as you see in the middle, is a molecule, and NAD and NADP, so the phosphorylated form of NAD, they are involved in multiple hundreds of reactions in your body. So this serves energy metabolism, but also biogenesis and so on. And in addition, NAD can also function as a signaling molecule through sirtuins, for example. These are deacetylases of proteins, or PARPs, which helps your DNA repair. So together, NAD is a very important cofactor for redox homeostasis, inflammation, calcium homeostasis, energy metabolism, DNA repair, and also mitochondrial biogenesis. However, similar to mitochondria, if we age, also NAD goes down. And this is dependent a bit on which organ, so it's not like everywhere at the same speed. And this can already start in your mid-30s or even a bit earlier. So to counteract an NAD decline and to support mitochondrial health, NAD boosters were considered. So you can either boost NAD levels by inhibition of the degradation of NAD, or you can boost NAD biosynthesis. And in particular, you have three canonical pathways how you can give NAD precursors to boost NAD levels. So these are the salvage pathway in blue. Then you have the Price-Handler pathway in green, and the de novo biosynthesis in gray. So each of these three pathways can be supplemented. So you have, for example, tryptophan is for the de novo biosynthesis. Then you have nicotinic acid as a substrate for the Price-Handler pathway. And then you have NMN, NR, and NAM that could feed the salvage pathway. Tryptophan has many functions. It's just an amino acid, so it's also used for proteins, and the way to NAD is rather long in terms of reactions. And nicotinic acid is known to have side effects, especially at higher doses like skin flushing, and at extreme high doses you can also have an impact on the liver. The blue ones, so NMN, NR, and NAM, they are considered safe, and so you can use them at high doses, and so we focus on these three. So each of them has been shown to increase NAD, but what was missing so far was a head-to-head comparison. So is one better than the other or not? So therefore, we designed a clinical trial in healthy population. So healthy population means we have people in the mid-30s and the BMI around 22. And these people consumed either NR, NMN, NAM, or placebo over 14 days. And there we were looking into a so-called chronic effect. So do we have a change in baseline at day 0 compared to day 14? And at the acute effect. So directly after you consume the precursor, does it reach your circulation or not? Generally, we took blood and urine samples as a readout, so we don't have biopsies for organ comparisons. So going first to the chronic effect, so what you can see in the middle is on the y-axis you have the NAD concentrations that we reach in the blood. On the x-axis you have the different the different treatments. And what you nicely see is that placebo and NAM do not affect baseline concentrations of NAD. However, both NMN and NR lead to a twofold increase of NAD levels in whole blood after 14 days of consumption. So what we can say here is basically NR and NMN are comparable, and they lead to a sustainable increase of NAD levels in your circulation. On the opposite, if we look into the acute effect at the first visit 3 and at visit 4, so what happens over 4 hours when you eat the different supplements? You see that the placebo does not have an effect as expected. NAM at both visits increases NAD, though not to a very big extent. And NR and NMN hardly have any effect. So we were further looking, okay, NMN and NR don't have an effect, so are they themselves actually absorbed? And what you can appreciate on the right side is if we look into the precursor parameters, if we give NAM, you have a huge increase in NAM. You have a little bit of increase in NR and NMN, which means the NAM transiently boosts the salvage pathway. However, if we look into NR and NMN, we basically don't have any change, not even when they are given themselves. So for us, this means, okay, probably NR and NMN are not or slowly absorbed, and they might be prone to gut metabolism or affect the gut itself. So to look a little bit into this, so we didn't have fecal samples, we chose an ex vivo approach. So we took the microbiota from humans, so directly primary microbiota, and then we incubated this microbiota with the different NAD precursors in microbial cell culture. We did this for healthy population, but we also took the microbiota from healthy elderly and from IBD patients where you expect some kind of dysbiosis. And what you can see on the right side is if we look into NR, we see that over time NR gets degraded, so it goes down. And then below, at the same time, you see that you have an increase of nicotinamide. It's transient. and nicotinamide is then further deamidated to nicotinic acid. So this deamidation effect we also see with nicotinamide itself, but what we know is nicotinamide gets readily absorbed, so it probably never reaches the gut. Besides this kind of NAD metabolism in the microbiome, which we also see with the NMN treatment, we were also looking, is the microbiome itself affected by NR and NMN? And what we nicely see is if we look into bacterial cell density, after 48 hours we have a higher bacterial cell density, but we also have a higher concentration of short-chain fatty acids, and short-chain fatty acids are known to positively affect the immune function and the gut barrier function. So you might have a dual effect besides the known NAD boosting effect. Then finally, we know now, okay, if you have microbial metabolism, you have nicotinic acid and you have nicotinamide, so you have basically substrates for price handler pathway, but also for the salvage pathway at the same time. So then we were thinking, okay, what is now increasing our whole blood NAD levels? So we took fresh whole blood and incubated it over 7 hours with different NAD precursors coming from the salvage pathway, NMN, NR, and NAM, or coming from the price handler pathway, so NAMN, NAR, and nicotinic acid. And what you nicely see is that in whole blood, nicotinic acid and NAR nicely increase NAD over 7 hours, which is pretty interesting because commonly you eat these salvage pathway precursors, but your whole blood effect is finally coming from the price handler pathway. So finally, we came up with a model where we proposed that NAM has basically an acute effect. It is readily absorbed in the gut. It increases its own level in whole blood, and it increases also NAD concentrations. But it's also quickly metabolized to the degradation products and then secreted in the urine. On the other hand, we have the chronic effect of NR and NMN. So we have a low absorption or no absorption. Then we have microbial hydrolysis and deamidation, which leads to NAM and nicotinic acid. Both of them could be absorbed. The latter gives rise to NAD. And then after organ metabolism, you of course also have some degradation products and secretion in the urine. So this already brings me to the conclusion. I think we have a new piece of insights that deepens our understanding of how different NAD precursors compare and act. So we know after 14 days, NR and NMN lead to a sustainable increase of NAD concentrations in humans, while NAM does not. And we have evidence that NR and NMN actually act as slow release NAD boosters since they are retained in the gut. And third, or fourth in this case, we have evidence that the NAD precursors themselves also affect the human microbiome, and this could support the production of healthy metabolites. So finally, since NR and NMN are potent precursors, we believe that they can truly support cellular energy, mitochondrial function, and immunity, and that the microbiome plays an important role in their modes of action. So the whole story, if you want to have more detailed, is open access, published in Nature Metabolism. With the QR code, you can easily access the publication. And with this, I want to thank Nestlé Research, so mainly the Nestlé Institute of Food Safety and Analytical Sciences, and the Clinical Research Unit who did the clinical trial. then Nestlé Health Science and Cryptobiotics for the microbial experiments. Yes, and that's it.